it is my absolute pleasure to introduce our speaker today, Dr. Adam Avery. Dr. Avery has an undergraduate degree in biochemistry from Oakland University. He earned his PhD in biological chemistry at the University of Michigan. As a graduate student in Anne Vojtek's lab, Adam gained expertise in protein-protein interactions in experimental approaches, including yeast-two hybrid. As a graduate student, Adam was a fellow on an NIH Cellular Biotechnology training program. Following the completion of his PhD, Adam worked in the biotech industry, where he developed cell-based assays for primary and secondary drug screening. Subsequently, he performed postdoctoral studies at Thomas Hayes' lab at the University of Minnesota. As a postdoc, Adam commenced his studies of the molecular and cellular mechanisms underlying SCA5. This work combines protein biochemistry and genetic and cell biological approaches in the model organism Drosophila. Adam also developed a patented cell-based assay to identify small molecule modulators of beta-III-spectrin, the SCA5 disease protein. Adam is currently an assistant professor of biochemistry at Oakland University. His research program reflects his broad training in biochemistry, genetics, and drug screening. Adam's lab has continued to make important contributions to our understanding of SCA5 disease mechanisms. Further, his research lab has contributed to a collaborative SCA5 drug discovery project. So with that, I'll turn the floor over. Dr. Avery, the floor is yours.
Okay, can you hear me?
Yes, we can.
Okay, Celeste, thank you for that introduction. Again, my name's Adam Avery. I am a basic science researcher, unlike Teresa Zysowicz, who talked on SCA5 a couple weeks ago, who's a neurologist and sees SCA5 patients. I hope that this talk kind of dovetails with her discussion of especially some of the clinical features of SCA5 disease. But as a basic science researcher studying SCA5, when I think of SCA5, I think about it really from a molecular level. So how, for example, do SCA5 mutations in the SCA5 disease protein beta-III-spectrin impact the structure of beta-III-spectrin, or the how do those mutations impact the binding of beta-III-spectrin to other proteins? And I also think about SCA5 at a cellular level. So how do mutations in the beta-III-spectrin protein impact the function of beta-III-spectrin to support neuron structure and function? And as Celeste mentioned, over the last about 5 years, I've also been a part of a SCA5 therapeutic discovery project in collaboration with investigators at the University of Minnesota, where I did my postdoc. And in this webinar, I'm going to touch on both the therapeutic discovery side of my work, but also our work into SCA5 disease mechanisms. All right. I'm trying to advance my slide. There it is. Okay, so just again, an overview of my lab's interests, my personal interests. These are the three kind of broad areas of our research focus. So again, we're interested in understanding the molecular and cellular mechanisms that underlie SCA5 pathogenesis, and we are also over the last 5 years have been working on an SCA5 therapeutic project, again with collaboration, and this is a collaboration with the University of Minnesota. And I'm also interested in understanding the basic biology of beta-spectrin, so the basic cellular and molecular biology of beta-spectrin in the absence of SCA5 mutations and healthy tissue, because I believe that an understanding of really a rigorous understanding of how beta-III-spectrin supports functions in neurons is interesting in itself, but it's really critical for developing or understanding SCA5 disease mechanisms. So a little bit about the biology of SCA5. This disorder causes disability through ataxia of the lower and upper extremities, slurred speech, uncontrolled eye movements. Age of onset is greatly variable. Initially, SCA5 was considered an adult onset disorder with symptoms starting in 20s or 30s on average. But many SCA5 mutations have now been reported, and there's probably an equal number of mutations that are associated with early onset SCA5 with these symptoms starting in infants or adolescents. And in this case, in early onset SCA5, this is a little bit different disease in that the symptoms are more severe. That include developmental delay, and they can be associated with cognitive deficits. The cognitive deficits, that's interesting because it suggests that there might be other parts of the brain that are impacted by these mutations outside of the cerebellum. However, a hallmark of SCA5 is atrophy or neurodegeneration of the cerebellum. That's especially the case in adult onset cases. However, in these early onset SCA5, it could also be viewed as a neurodevelopmental disorder, and there's probably a defect in the development of the cerebellum. So on right are two MRI images. You saw images like these in the talk a couple of weeks ago by Dr. Zeisowicz. But this is an MRI of a non-SCA5 patient. The red box is the cerebellum. You can see that it fills up this space in the hindbrain. However, this SCA5 patient, the cerebellum looks a little bit smaller. There's more black space around the cerebellum, which is indicative of atrophy of the cerebellum. So that's a hallmark of SCA5 is atrophy of the cerebellum. The primary neuronal target in SCA5 pathogenesis is considered the Purkinje neuron or Purkinje cells. This is autopsy tissue down here of a non-SCA5 patient. So in brown is a Purkinje neuron. So this is the cell body, and these are the dendrites that are extended by this cell body. You can see that there's an elaborate dendritic arbor. There'll be better kind of higher resolution images of Purkinje neurons to come in this talk. But on the right, in the SCA5 patient, here's the cell body. Here are the dendrites. You can see that the region of the cerebellar cortex that contains these Purkinje cell dendrites is a lot thinner compared to the control. And clearly, this Purkinje neuron does not look as healthy as the one here. So this suggests that one impact of SCA5 mutations is to cause a loss of Purkinje cell dendritic arborization. So likely arbor defects in Purkinje cells is a component of the pathogenic mechanism. This disease is autosomal dominant. This means that the mutations are on an autosome, not a sex chromosome, and dominant means that you only have to inherit one copy of the beta-spectrin gene that has a mutation in order to develop the disorder. That gene, again, is SBTBN2, and that encodes a cytoskeletal protein called beta-III-spectrin. All right. Generally speaking, the functional unit of a beta-spectrin is considered to be a heterotetramer. So in this cartoon, the heterotetramer is composed of two units of beta-spectrin, which are these two blue noodles, and two units of alpha spectrin, which are the two green noodles. And then each end of the heterotetramer through the beta-spectrin units are bound. Each end of the heterotetramer is bound to a short actin filament. And this, in this way, the heterotetramer cross-links actin filaments and forms a spectrin actin cytoskeleton or mesh that underlies the plasma membrane. So this spectrin actin mesh lines the inner leaf of the plasma membrane. There's an important adapter protein that binds beta-spectrin called ankyrin, and through ankyrin, the spectrin cytoskeleton is linked to membrane proteins such as transporters, so ion channels, amino acid transporters, or cell adhesion molecules. And in general, the spectrin cytoskeleton is thought to confer shape and mechanical resiliency to the plasma membrane, and also to stabilize and organize membrane proteins such as transporters, cell adhesion molecules. Beta-III-spectrin is predominantly expressed in the brain. So this is a slice of a rat brain, and here the rat brain has been stained for beta-III-spectrin mRNA. So this staining is purple, where you see more intense purple staining. That's where beta-III-spectrin expression is higher at the mRNA level. And so one thing that you can see is that beta-III-spectrin is expressed in different regions of the brain. For example, you can see some purple staining up here in the cerebral cortex, but its expression in the cerebellum really stands out. And within the cerebellum, beta-III-spectrin is protein is expressed in Purkinje neurons. So the green signal here represents beta-III-spectrin protein. These are Purkinje neurons. These are the cell bodies, and Purkinje neurons, and you can see that beta-III-spectrin is present throughout the dendritic arbor. One interesting thing, kind of fact about beta-III-spectrin is that it specifically localizes to the soma and dendrites of Purkinje neurons. It's absent from the axon, where other forms of beta-spectrin, such as beta-2 or beta-4, are present. Okay, but beta-III-spectrin is highly expressed in Purkinje neurons, and it's present in the soma and throughout the dendritic arbor. All right, and it's known in part from studies in mice that beta-III-spectrin is required for Purkinje cell dendritic arborization. So there have been 2 beta-III-spectrin knockout mice generated. A beta-III-spectrin knockout mouse is a mouse in which the beta-spectrin gene has been deleted. So in the knockout mouse, beta-III-spectrin is not expressed. And so this image here comes from one of these papers. This is a Purkinje neuron in a wild-type mouse that has beta-III-spectrin expression. This is so cell body, this really pretty elaborate dendritic arbor. This is at 5 to 6 weeks, so an early time point, and then this is the neuron at a later time point. And this is in the beta-III-spectrin knockout mouse. So even at this early time point, you can see that in the absence of beta-III-spectrin, the arbor clearly looks frayed compared to the control neuron. And by the later time point, there is really a strong loss of arborization. There's a strong atrophy of the arbor. So this is one piece of data that supports that Purkinje beta-III-spectrin function is required for Purkinje cell dendritic arborization. And there was another interesting observation that came from these beta-III-spectrin knockout mice. This is looking at, which is the mislocalization of postsynaptic proteins. And so these studies looked at 2 proteins. One is a glutamate receptor, and the other is a glutamate transporter, EAAT4. I'll talk a little bit more about this transporter later in the talk. All right, but in wild-type healthy Purkinje neurons that contain beta-III-spectrin, the glutamate receptor, which is the red or yellow signal, it is present in these puncta that are present along dendrites. These puncta are sites of synapses. So this glutamate receptor is important and it plays an important function in synapses. So those are sites of communication with other neurons. All right, but in the absence of beta-III-spectrin, the glutamate receptor starts to, instead of, it's mislocalized, it accumulates in the cell body. So this yellow signal in the cell body, it accumulates in the cell body where it's not present in the wild type neurons. And this suggests, so this glutamate receptor should be out in the dendrites at sites of synapses where beta-III-spectrin normally localizes. And this supports the general role that beta-III-spectrin, like other forms of spectrin, have this role, this functional role to stabilize membrane proteins at sites of synapses, at the plasma membrane. In this case, postsynaptic proteins involved in glutamate signaling at synapses located in Purkinje cell dendrites. Okay, so again, SCA5 stems from mutations in the SBTBN2 gene encoding beta-III-spectrin. This is a domain diagram of beta-III-spectrin. This is the N-terminus of beta-III-spectrin. This is the C-terminus. At the N-terminus, there's a short 50 amino acid amino acid sequence that leads into two subdomains called CH1 and CH2. CH stands for calponin homology. And together, this N-terminus and the two CH domains make up what's called the actin binding domain of beta-spectrin. The actin binding domain is followed by 17 spectrin repeat domains, and then there's a C-terminal plextrin homology domain, or pH domain. The pH domain binds phosphoinositide lipids, and is probably important for mediating the, or at least contributes to the association of beta-III spectrin with membranes within the cell, such as the plasma membrane. Okay, and it's known that spectrin repeat domain 1 and 2 is important for nucleating the interaction of alpha-2 spectrin, of beta spectrin with alpha-2 spectrin, and that repeat domains 14 and 15 bind the protein ankyrin. Again, ankyrin is this adapter protein that can link spectrin to membrane proteins such as maybe glutamate receptors. All right, and shown are the first 3 SCA5 mutations that were identified in beta-III spectrin. These mutations were reported back in 2006 from Laura Ronham's lab. 2 of these mutations are small end-frame deletions in the 3rd spectrin repeat domain. This E532M44 delete mutation is the American mutation, or Lincoln mutation, as it's sometimes called, that localizes the repeat 3. And then L253 is a leucine amino acid substitution for a proline residue, and that's localized to the actin binding domain. And I'll talk more about these mutations later in the talk. Okay, so general approaches to studying SCA5, and I've broken this down into 2 groups. 2 categories. One is biochemical or biophysical approaches to understand how SCA5 mutations impact beta spectrin structure or its interaction with other proteins. And the second is animal or cellular models to study how SCA5 mutations impact the function of beta-III spectrin to support neuron structure and signaling. So I'll begin with this biochemical, biophysical approach. Okay, so in general, a protein in a cell does not function in isolation by itself. Often, a protein requires interaction with other proteins to perform its function. And the identity of those other proteins, those interacting proteins, give information on the function of your protein of interest. So, for example, beta-III spectrin. So it is known, for example, as I just talked about, that beta-III spectrin binds actin, alpha spectrin, ankyrin. There are some additional proteins that are known to bind a beta-III spectrin as well. But likely, our understanding of all the different proteins that beta-III spectrin interacts with is incomplete. And we really need to, in my opinion, a rigorous understanding of the beta-III spectrin protein interactome, not just to understand beta-III spectrin function, but to have a more complete understanding of SCA5 disease mechanisms. All right, so I'll go through So how do you identify novel protein interactors for a protein of interest, such as beta-III spectrin? One approach is called the yeast-two hybrid screen. This is an approach that I'm well familiar with because I learned how to do it as a graduate student. And this is so I'm giving an example of a yeast-two hybrid screen that was performed by Mandy Jackson. She's a researcher that still continues research on SCA5. And so Mandy Jackson was interested in this protein EAAT4, which I just mentioned previously in the beta-III spectrum knockout mouse was mislocalized in that knockout mouse. This is so excitatory amino acid transporter 4. It's a glutamate transporter. It's important for removing glutamate from the synaptic cleft. So she was interested in identifying proteins that interact with EAAT4. And so she performed a yeast-two hybrid screen. Again, this is a protein-protein interaction screen. and to identify novel interactors for EAAT4. So in a yeast-two hybrid screen, your protein of interest, which in this case for Manny Jackson was EAAT4, is called the bait. And you screen this bait protein against a library of other proteins. And if any of the proteins in that library interact with EAAT4, that is going to allow yeast to grow on selective media. All right. And so this is a protein-protein interaction screen. It's not super relevant that it was performed in yeast. But what's important was what she found in this screen. So she found that there are two proteins. She found two novel protein interactors for EAAT4. One of these proteins was a regulator of the actin cytoskeleton, a protein called a ROGEF. The other protein was beta-III-spectrin. And specifically, she found that a C-terminal region of beta-III-spectrin containing repeats 14 through 17 bound to EAAT4. So this actually is the region of beta-III-spectrin that also interacts with Actin. So why is this important? The identification of this, trying to get my slide to move forward. Yes. This important was this identification of this novel beta-III-spectrin interactor EAAT4 was one of the first pieces of evidence that suggested a functional role for beta-III-spectrin in regulating glutamate signaling. Okay, so that protein-protein interaction gave insight into the function of beta-III-spectrin in Purkinje neurons, or at least one of its functions. My lab has also been interested in identifying novel beta-III-spectrin interactors, and we also performed a yeast-two hybrid screen recently. This work is not published, and so we used beta-III-spectrin as a bait in our yeast-two hybrid screen. We didn't use the full-length protein, but instead we used the N-terminal half, so the N-terminus through spectrin repeat 7. And our reasoning for using this N-terminal half is because this is where all the SCA5 mutations cluster. So we were interested in identifying proteins that bound this region with the idea that maybe some of these novel protein-protein interactions are disrupted by SCA5 mutations. Okay, and so we did this screen. We used the N-terminal half of beta-III-spectrin, and we screened a library of proteins from adult human brain, and we identified about 20 novel beta-III-spectrin interactors, which are shown here. One of the interactors that we identified was not a novel interactor, is actually alpha-2-spectrin, a well-established interaction. And the interaction of alpha-2-spectrin with beta-III-spectrin in this yeast-two hybrid system allows yeast to grow on selective media. So these white dots are actually yeast colonies, and they wouldn't be able to, these yeast would not be able to grow on this minus Walk media if beta-III-spectrin was not interacting with alpha-spectrin. All right, another protein that we're interested in that came out of this screen is one called MACF1. This is a giant cytoskeletal protein. It has some similarities to beta-spectrin. The interaction between beta-III-spectrin and MACF is robust. And this protein is interesting because it is known to be required for dendritic arborization like beta-III-spectrin. It's a disease protein. And its mutations in MACF also cause atrophy of the cerebellum. So this is kind of ongoing work in my lab based on our yeast-two hybrid screen. And we're interested in whether this interaction between beta-III-spectrin and MACF contribute to SCA5 pathogenesis. Now, there are a lot of different proteins that came out in this screen. Some have functions that perform functions in cells that have not currently been linked to beta-III-spectrin. So we think it's possible that beta-III-spectrin might have a really diverse set of functions it performs in cells through by performing or scaffolding interactions with different protein complexes. Okay, so that's ongoing work, and that hasn't been published. And now I want to talk a little bit about some of the work that we've been doing over the past 5 or so years. Some of this has been, or most of this has been published. And this work has the question that this work has tried to address is whether SCA5 mutations, including this leucine 253P mutation, disrupt the binding of beta-III-spectrin to some well-established interactors, including alpha actin and alpha-2 spectrin. We have not asked how SCA5 mutations impact the binding of beta spectrin to ankyrin, for example, because as far as I am aware of, there's no SCA5 mutations that have been reported near the ankyrin binding site. All right. Okay, so the L253P mutation, again, is a substitution of a leucine residue for a proline residue. And this mutation localizes to the CH2 domain, which is a component of the actin binding domain. So we wanted to know whether this mutation disrupts the function of beta spectrin to bind actin. And so to gain insight in how that mutation impacts the structure and function of the actin binding domain, we generated what's called a structural homology domain. So this is a predicted structure of the actin binding domain. Again, it contains two subdomains, CH1, which is the left lobe of this structure, and CH2, which is the right lobe of this structure. And leucine 253 is this magenta residue that is in CH2. And it's also noteworthy that this leucine residue is at the interface of CH2 and CH1. All right, it's actually predicted to form contacts in CH1, and through those contacts may bridge the two CH domains together. So we wanted to know how the L253P mutation at this site impacts the function of the ABD to bind actin. And so we performed a type of actin binding assay that's called the actin co-sedimentation assay. So in this assay, a fixed amount of the beta spectrin ABD is mixed with an increasing amount of actin. And this data is for the wild type ABD. And the y-axis is the fraction of ABD that is bound. And as you can see, as the concentration of actin filaments is increased, there's a progressive increase in the amount of the wild type ABD that is bound to the actin filaments. And this data can be fit with a binding curve, which is this black line, to solve for what's a value called a KD, which is a dissociation constant. And a dissociation constant is a quantitative measure of the affinity of two proteins. And so the KD for the wild type ABD, actin binding domain, is a 75 micromolar. All right, a 75 micromolar KD is a low affinity protein-protein interaction. So this data shows that the ABD of beta-III spectrin binds actin filaments with low affinity. However, when we repeated this assay for the L253P ABD, what we observed is that the mutant ABD was entirely bound to actin at every actin concentration tested. So this suggested to us that the mutation was causing a large increase in actin binding affinity. And using lower reactant concentrations and different curve fitting, we were able to generate an estimate of the KD for the mutant, which is 75 nanomolar. All right, so the KD of the mutant is 75 nanomolar. This is 1,000-fold lower than the KD of the 75 of the wild type protein, which is 75 micromolar. So in other words, the mutation is causing about a 1,000-fold increase in actin binding affinity, and that's a huge effect. And we proposed a model in which the actin binding domain exists in two conformations. One conformation, the CH1 and CH2 domains are closely opposed, and this is mediated by leucine 253 and its hydrophobic contacts. And the other conformation, which we call the open conformation, the two CH domains are spatially separated. And we think it's this open conformation that binds to actin filaments. We proposed that these two conformations were in equilibrium and that the L253P mutation was shifting this equilibrium towards the open state. Okay, we were able to confirm this model by a couple different biophysical approaches. One was an approach called cryo-electron microscopy. And through cryo-EM, we were able to generate an atomic model of the L253P ABD bound to an actin filament. So here, the actin filament is blue, and the L253P ABD is magenta and red. Now, again, the ABD contains two CH domains, CH1 and CH2. But there are two important pieces of information came out of this model. One is, in this model, only a single CH domain can be observed bound to the actin filament. So this is one of the CH domains, and it's bound to the actin filament. And you cannot actually see the second CH domain in the structure. What that means is that that second CH domain is not tightly associated with the CH domain that's bound to actin. It also means that that second CH domain is not bound tightly somewhere else on the actin filament. Likely, that second CH domain is flopping around in solution and can't be resolved in this structure for that reason, because it's not fixed in a single position. And that supports that it's an open state of the ABD in which the two CH domains are spatially separated. That is the actin binding state. All right, the other important piece of information that came from this structural study was the presence of this red helix that leads into the globular fold of the CH domain that's bound to actin. The presence of this helix unambiguously identified the bound CH domain as CH1. And that means that high affinity actin binding caused by the CH2 localized L253P mutation is directly mediated by CH1. So the mutation in the CH2 domain is not causing high affinity actin binding by, for example, exposing or generating a de novo high affinity actin binding site in CH2. Okay, so I just talked about the L253P mutation, which was the first SCA5 mutation identified within the actin binding domain. But since the report of that mutation back in 2006, at least 9 more SCA5 mutations have been reported that all localized to the actin binding domain. So we wanted to know how these mutations, whether these mutations also impact the binding of beta-spectrin to actin. And what's really striking is that all of these SCA5 mutations cluster at the interface of CH1 and CH2, just like L253. So right off the bat, that suggested to us that these mutations were likely going to have a similar impact on actin binding as the L253P mutation. So to test this, we purified the ABDs, the wild type ABD or the beta-III-spectrin ABDs containing the different mutations, and performed actin co-sedimentation assays. And so these are the binding curves for the binding data from the co-seds. And this black curve at the bottom is the binding curve for the wild type protein. And the brown curve all the way at the top is the binding curve for the L253P ABD. And as you can see, the binding curves for all the other mutations fall above the wild type curve and below the L253P curve. So what this means is that all of these mutations increase actin binding relative to wild type, although none increase actin binding quite as high as the L253P mutation. So all of these mutations, all of the mutations that we have tested so far within the ABD cause increased actin binding. And this has, I believe, an important therapeutic implication because it suggests that if you can identify a small molecule drug that can bind one of these mutant beta-III mutant beta that can specifically bind mutant beta-III spectrin and reduce its affinity to actin, then that drug might be broadly useful as a SCA5 therapeutic. Okay, now I'll next talk about the second experimental approach, which is the use of animal or cellular models. Okay, so one of the model systems that we work with in the lab is the fruit fly, Drosophila melanogaster. And fruit flies are really popular in biomedical science for understanding both the basic biology of proteins, but also disease mechanisms that might be conserved between flies and humans. And one of the reasons that fruit flies are a popular model system is because most human protein coding genes are conserved in flies. So for example, human beta-III-spectrin, it's has a similar protein. It's conserved in flies. In flies, the fly homolog of human beta-III-spectrin is simply termed a beta-spectrin. Fruit flies, there are numerous genetic tools that allow you to do some really advanced experiments. They also have a short life cycle. They're inexpensive to work with, which means that experiments can be done more quickly and less expensively than they can be done in higher order animals such as mice. So this is the fruit fly life cycle. Flies will start out as an egg. A larva will hatch from the egg, and then this larva will go through a couple rounds of metamorphosis to pass from the first instar larva to the second instar larva to what's called a third instar larva. This is the largest, the third instar larva are the largest larva, and this is actually the developmental stage of fruit flies that we do a lot of our studies. The larva will then form a pupal case, and then adults will close from the pupal case. And at room temperature, this takes about 10 days. Okay, so one thing that we use fruit flies for is to understand how SCA5 mutations impact the formation or maintenance of complex dendritic arbors as extended by Purkinje neurons. Except flies don't contain Purkinje neurons, but they do contain a neuron that's called the class 4 dendritic arborization, or DA neuron, that extends a complex arbor like Purkinje neurons. So this is an image of a class 4 DA neuron. In the center is the cell body, and basically every black line that you see within this field of view is a dendrite that is extended from this cell body. So this neuron has a very complex dendritic arbor. Another neat feature of this neuron is that these neurons live the larval epidermis. And so this is a third instar larva that's been kind of highly magnified. This third instar larva is about probably 6 millimeters in length, actually. So these neurons line the larval epidermis, and the larval epidermis is semi-translucent. So that means if you express a fluorescent protein such as GFP in these neurons, which was done here, then you can image these neurons in a fully intact living animal. And that's important because then you can study these neurons in their native environment with all their normal cell connections. Okay, so one of the experiments that we did was to introduce the L253P mutation into fly beta-spectrin, and then we expressed this L253P or SCA5 beta-spectrin in class 4 DA neurons. And as you can see, this expression of this mutant beta-spectrin in this neuron results in a really striking arbor phenotype. The arbor looks small or shrunk compared to the control neuron expressing wild-type beta-spectrin. And we know that this small arbor phenotype reflects two processes. One is a degenerative process in which distal dendrites are progressively lost. And another is a developmental defect in which new dendrites are formed, but they're only formed in areas close to the cell body. Okay, we also asked how does wild type and mutant beta spectrum localize within the dendritic arbor? So this data is for the wild type beta spectrum. You can see that it's present in the cell body, and it's present throughout the dendritic arbor. Here, the dendritic arbor is counter-labeled with a red fluorescent membrane protein. In contrast, the mutant beta spectrum is lost from distal dendrites, and instead it accumulates in the proximal dendrites and in the cell body. So here is actually you can see the cell body here and here under different contrast settings, so you can appreciate the accumulation of the mutant beta spectrum in the cell body relative to wild type. So conclusions from these studies in flies. One, it shows that the L253P mutation that causes high affinity actin binding is associated with a redistribution of spectrin from distal dendrites to the proximal dendrites into the cell body. Another thing and data I didn't show was that this the mutant beta spectrin dominantly mislocalizes interacting proteins such as alpha spectrin. So alpha spectrin also accumulates in the cell body. And another important takeaway from this project is that the loss of spectrin in the distal dendrites is associated with the degeneration of distal dendrites. And that suggests that a normal function of beta spectrin in the dendritic arbor is to stabilize distal dendrites. So we think that beta spectrin needs to enter into these distal dendrites and stabilize them to prevent their collapse or degeneration. Okay. Now I'll shift to mouse models. So mouse models are widely used in ataxia research. And there are two SCA5 mouse models that have been developed. The first one was published back in 2014 from Laura Ronham's lab, and this was on the American or the Lincoln mutation. Okay, so this is the paper that reported on this American mouse model. This is what's called a transgenic mouse, which is expressing human wild type beta-III spectrin or human beta-III spectrin containing the American mutation. And these wild type and mutant beta spectrin transgenes are specifically being expressed in Purkinje neurons. And what was found in this study was that expression of the mutant beta spectrin in Purkinje neurons led to cerebellar degeneration and associated motor deficits. Another important takeaway from this paper is that they observed that a glutamate receptor, the metabotropic glutamate receptor 1 alpha, was mislocalized. So normally this glutamate receptor localizes to dendritic spines, which are the sites of synapses between Purkinje neurons and other neurons. And this mislocalization of the mGlu-R1 receptor was associated with defects in mGlu-1R's alpha signaling. And this is important because it was really the first in vivo study that linked an SCA5 mutation, in this case the American mutation, to defects in glutamate signaling. Okay, the second SCA5 mouse model that has been generated hasn't been published yet. This is the L250. This is a SCA5 mouse is for the L253P mutation. And this mouse was generated in Harry Orr's lab. And this has been a collaborative project between Harry Orr, Tom Hayes, who is my postdoc advisor, and myself. And most of the work that I'll be presenting was generated by talented senior researcher Brennan O'Callaghan in Harry Orr's lab. This is a knock-in mouse, and that means that CRISPR-Cas9 was used to introduce the L253P mutation into the native mouse beta-III spectrin gene. Okay. And just waiting for my slides to advance. Let's see. Okay, so importantly, this mouse shows an ataxic phenotype. It's a mild phenotype, but it's an ataxic phenotype nonetheless. And so the phenotype it shows is a foot slip phenotype. So when mice are asked to cross a small beam connecting two platforms, mice containing the L253P mutations show an increase in the number of foot slips. All right, so there's this motor phenotype that is associated that we've identified in this mouse, and it's this foot slip phenotype. And we can see this phenotype in 12-week-old mice, or and even older 20-week mice. All right, we also have performed immunohistochemistry to look at where beta-III-spectrin localizes within the wild type and the LT53P mutant mice. And so here, this shows cerebellar Purkinje neurons. In this case, the blue signal here is staining for a Purkinje neuron protein called calbindin. So calbindin, you can see it in the cell body of these Purkinje neurons and also in the dendrites. And the red signal is for beta-III-spectrin. And again, this is in the wild type mouse that doesn't contain the mutation. This panel down here shows just the beta-III-spectrin signal. So you can see beta-III-spectrin is present in the cell bodies and throughout the dendritic arbor. And this localization of beta-III-spectrin is perfectly consistent with all prior reports on where beta-III-spectrin localizes in Purkinje neurons. We've also looked at the localization of F-actin since beta-III-spectrin binds F-actin. And we can see that F-actin is present just about everywhere in the region of the cerebellum that contains Purkinje cell dendrites. And it's also present in the cell body, although it's difficult to see the F-actin signal here because of the strong beta-III-spectrin signal. Okay, we then probed beta-III-spectrin localization in mice that are heterozygous for the LT53P mutation. So these mice contain one wild type beta-III-spectrin gene and one LT53P gene. And we observe a defect in the localization of beta-III-spectrin in these mice. So what we observe is that beta-III-spectrin begins to be present on these small inclusions or puncta in the cell body. And the other neat observation is that beta-III-spectrin now accumulates at the plasma membrane of the soma and also outlines the plasma membrane of the proximal dendrites. So let me just skip back and look at, you can see beta-III-spectrin here. You don't see any beta-III-spectrin signal at the cortex or the plasma membrane of these wild-type Purkinje neurons. Or down here, you don't see this accumulation of beta-III-spectrin at the cortex, and there's very little evidence of puncta there. But here, now you see that this accumulation of beta-III-spectrin at the plasma membrane of the soma and also the proximal dendrites. And when you look at F-actin staining, it's a little bit hard to see in these, but everywhere that beta-III-spectrin localizes, it co-localizes with F-actin, including on these puncta and at the cell membrane of the soma. Okay, and then this is the localization of mice that are homozygous for beta-III-spectrin. So these mice contain 2 copies of the LT53P gene. So all beta-III-spectrin expressed in these Purkinje neurons are the mutant form. And now these puncta are more pronounced in the cell body. The staining at the cell membrane of the plasma membrane of the cell bodies is also stronger, and you can see really strong outlining of the proximal dendrites as well. And when you look at F-actin co-localization, F-actin again co-localizes with everywhere that you see beta-III-spectrin, including on these large puncta in the cell body. You see really intense co-localization with F-actin. And so conclusions from this are that the LT53P mutation is causing a redistribution of beta-III-spectrin from distal arbor to the proximal dendrites in the cell body. This is a very similar effect that we saw in our SCA5 fly model, this redistribution of beta-spectrin. The mutant beta-spectrin also shows robust co-localization with F-actin, which supports that increased actin binding is the underlying disease is the underlying molecular consequence driving disease for the L253P mutation. And importantly, this mouse has an ataxic phenotype at a fairly early age, 12 weeks, which makes this mouse, we think, useful for future testing of drugs. Okay, now I'll switch gears and talk about SCA5 therapeutic approaches. I'll start with just describe or going over the central dogma of molecular biology. So in molecular biology, information encoded in DNA, so for example, the DNA of the SBTBN2 gene, is transferred to messenger RNA, so beta-III-spectrin mRNA in this case. And this messenger RNA then directs the synthesis of protein, in this case, beta-III-spectrin protein. So for a therapeutic approach, you can possibly target beta-spectrin at the DNA level, the RNA level, the mRNA level, or the protein level. So here are some possible therapeutic approaches. One would be to correct the DNA mutation or variant in the SBTBN2 gene. And to do this would require CRISPR-Cas9 genomic engineering. Unfortunately, CRISPR-Cas9 engineering it has not been developed well enough yet that it could be used to treat an ataxia such as SCA5. But maybe in the future, this would be a viable approach. A second approach would be to target and degrade the mutant beta-III-spectrin mRNA. So target beta-spectrin expression at the mRNA level. And so this could be done through RNA interference or a technique called antisense oligonucleotides, or ASOs. And ASOs are actually being developed for the treatment of other types of SCAs, spinocerebellar ataxias. And I'll talk a little bit more about this on the next slide. Another approach would be to target or try to correct the aberrant activity of the mutant protein. And this could be by targeting the mutant protein with a small molecule drug. And this is really the focus of our therapeutic development, which I'll talk about in a few slides. Another general approach could be to target downstream effects of the mutant protein. So for example, if glutamate signaling is altered in Purkinje neurons due to mutations in beta-spectrin, maybe a small molecule drug could target a protein within the glutamate signaling pathway and help reverse some of the deleterious effects of that altered glutamate signaling. And then, as Dr. Zeisowicz had mentioned last week, physical therapy is also an option in order to improve motor coordination. And this is not an exhaustive list. This is a list that I came up with, but it's certainly not exhaustive. Okay, so this is a study that I came across just maybe a week ago, but it's relevant to SCA5 and beta-III-spectrin. So in this study, a group from Baylor College of Medicine in Texas was interested in developing antisense oligonucleotides, or ASOs, as a treatment for a disease caused by mutations in the SPT-AN1 gene. So the SPT-AN1 gene encodes alpha-2-spectrin. Alpha-2-spectrin is the protein that beta-spectrin forms a heterotetramer with. And so mutations in this gene encoding alpha-2-spectrin cause a neurological disorder called developmental and epileptic encephalopathy type 5. And so what they were interested in doing is developing ASOs that could specifically target the mRNA, the alpha-2-spectrin mRNA containing the mutation, but not target the wild type mRNA. And that is a challenge because the wild type mRNA and the mutant mRNA differ by only one base. So the ASO would have to have really specificity would have to be sensitive to just that single base change. But they were successful in doing this. They were able to generate several ASOs that caused degradation of the mutant mRNA and had little or very little effect on the wild type mRNA. And this is relevant to SCA5, not just because it's treating a neurological disorder caused by a mutation in an alpha spectrin gene, but because most SCA5 mutations are due to a single base change. So this kind of shows proof of concept that an ASO could be developed to treat, specifically degrade a beta-III-spectrin mRNA that differed from the wild type mRNA by just a single base. But really, currently, there's no, as far as I'm aware, I think this is a therapeutic option that should be explored, and hopefully there's some funds will become available to pursue ASOs for SCA5. Okay, so now I'm going to switch gears and talk about the drug discovery project that I've been working on in collaboration with Minnesota for about the past 5 years. And this is a project where we are trying to identify drug-like small molecules that specifically target the mutant beta-III-spectrin protein. And as I mentioned before, there are many SCA5 mutations that localize to the ABD, and all of these so far that we've tested cause increased actin binding. So our therapeutic strategy is to identify a small molecule that specifically targets the mutant beta spectrin, ABD, and reduces the binding of the mutant ABD, mutant beta spectrin to actin filaments. So we've developed an assay for drug screening. This is really a simple assay that contains just 2 components. One is an actin filament, which is this yellow tan protein. The actin filament is conjugated to a fluorescence dye called Alexafluor 568. And the second component of this assay is the L253 ABD, beta spectrin ABD, which is blue, and this ABD is fused to a green fluorescence protein. So this is a spectroscopic assay. It's a fluorescence assay, and when it's a FRET assay. So when you shine blue light on this protein complex, that excites the GFP protein, and because the GFP protein is within close proximity to the Alexafluor 568 dye that's attached to the actin filament, there's what's called a non-radiative exchange of energy from the excited GFP to the Alexafluor dye. That biophysical exchange phenomenon, this non-radiative exchange of energy, is called FRET. And this FRET is the signal that we are measuring in this assay. All right, and this assay has a really robust FRET efficiency, near 50%. So that's a huge FRET signal. And when we treat this assay with a compound called Swin halide A, which is an actin severing complex, it disassembles the complex and greatly reduces that FRET signal. So this is, and this data is after 20 minutes treatment with Swin halide A, but if you treat for 30 or 60 minutes, this FRET signal goes all the way down to 0. That means this assay has it has, number one, a very large FRET signal, and that FRET signal is entirely specific, meaning that the FRET signal is entirely a measure of the binding of the mutant ABD to actin. So we optimize this assay for high throughput drug screening. And then we use the optimized assay to screen a library containing 3,000 compounds that were FDA approved or approved for clinical trials. And we screen this library in duplicate. So these 2 scatter plots show the result of the duplicate screens. Each green dot represents a single compound, the effect of a single compound on the FRET signal after 30 or 60 minutes of treatment with that compound. And as you can see, there are some compounds that increase FRET relative to our control, what's called a DMSO control. And there are other compounds that decrease FRET relative to the DMSO control. So we were interested in compounds that decrease FRET relative to control because a decrease in FRET in this assay is interpreted as a decrease in binding of the mutant ABD to actin. And that's the activity that we're after. And so we went forward with the compounds that decrease FRET reproducibly between these two duplicate screens. These compounds were then repurchased to confirm their activity, and with the repurchased compounds, we repeated the FRET assay, this time generating dose response curves. So this panel of 16 shows the dose response curves for 16 compounds that had some of the larger effects. Here I'm highlighting one compound called mycofungin, which reduced the FRET signal by about 40%. We then wanted to confirm the activity of these compounds to reduce binding to mutant spectrin to actin using a different type of actin binding assay, what's called an orthologous assay. And we wanted this assay to be independent of fluorescence or FRET. And so the assay that we used is the actin co-sedimentation assay. And so we tested all the compounds that reduced FRET in these actin co-sedimentation assays. And most of the compounds that had activity in the FRET assay also had activity in the co-sedimentation assay. However, some compounds had kind of a strange effect in this co-sedimentation assay. And these compounds that had this strange effect, the strange effect was that they appeared to increase actin binding in this assay, which is opposite the effect of these compounds had in the FRET assay. And so we did some follow-up on these compounds that appeared to increase actin binding. So these are the compounds that have the green numbers by them. And what we found is that these compounds are actually aggregators. They're causing the ABD to aggregate, and that's a mechanism of action that we're not interested in. And so any compound that was having this aggregating effect, it was eliminated from our hit list. So that left us with about 9 compounds that we were able to confirm to have activity to reduce binding to mutant spectrin to actin in these co-sed assays. So this is an example of some of these compounds. Here is mycofungin, which reduced binding in the co-sed assay by about 50%. This is similar to the magnitude of the effect that this compound had in the FRET assay. Another compound here is tacrolimus. Okay, so these are the 9 hit compounds that came out of this screen of a 3,000 compound library. So these are the hit compounds that we were able to confirm activity in that orthologous binding assay. There are a couple compounds to me that are particular interest. One is mycofungin. This is because it had the largest effect. This is actually antifungal that can be injected for systemic fungal infections. Appears well tolerated at high dose. One challenge with this compound, so this is mycofungin, is that it's a very large compound, so there might be some trouble in it passing the blood-brain barrier. And then another is ginsenoside RB1. This compound had activity at very low doses, which is important for a drug. And this is actually a phytochemical that's derived from the ginseng plant. There's some interesting research papers on ginsenoside RB1. One that stood out to me was a study using a Parkinson's mouse model where treatment with this compound was able to reduce motor symptoms. Okay, so these are the 9 compounds currently that came out of this screen. I do want to just say that a lot of work has to be done on these compounds, further characterizing, for example, mechanism of action. Some of these compounds, we have to increase the potency so they have activity to reduce binding to mutant spectrin at lower concentrations. None of these compounds have been tested for treatment of SCA5 at this point. So there's no safety profile for these compounds, for example, in treating SCA5. So a lot of work yet is left to do on these compounds, but these are compounds that we are following up on. So in summary, we've developed a robust FRED assay for drug screening from a relatively small 3,000 compound library. We were able to identify 9 hits with with desired activity and confirmed activity to reduce binding of mutant spectrum to actin. And importantly, this establishes that the binding of spectrin to actin is an interaction that can be druggable. It can be modulated by small molecules. And next steps, for these 9 hit compounds, we want to test structural analogs to see if, so a structural analog of, let's say, the compound mycofungin is a compound that is very similar in structure to mycofungin, but differs just a little bit in its structure. So we want to basically see if we can slightly modify the structure of some of these compounds to improve the properties of the compound. So, for example, increase potency. Some of these compounds we'd like to see larger effect size, and we'd like to see these compounds have activity at lower concentrations. We screened a 3,000 compound library, but now we would like to use this assay to screen a 50,000 or 100,000 compounds in order to increase the number and structural diversity of hits. This is important because the more hits that we have to start with, the more likely it is that one of these molecules will pass through all the downstream tests that are required for approval of a therapeutic. And now we have this SCA5L253P mouse, which is just waiting for us to decide on some top hits to test within this mouse to see if they can alleviate the ataxic phenotype of the mouse, or even this mislocalization of beta spectrin that seems to be actin dependent within Purkinje neurons. Okay, so I'll just end on this acknowledgement slide. My lab, I have 2 really talented graduate students, Sarah Denna, who is pictured here. She is looking for a postdoc. She wants to continue in the ataxia field. Alexandra Atang is starting in medical school in the fall. And I've also had some other talented students contribute to this work. Dave Thomas lab, especially Robin Rebick and Piela Guhatha-Kurta, have been really instrumental, just critical process of the drug screening that we've done. They have a specialized FRET plate reader that allows us to screen like a 50,000 compound library in just a couple days. And only this lab has that technology. So this has been a really great collaboration. Tom Hayes, my postdoc advisor at Minnesota, has been involved in this drug screening project from the start. He was the PI on our first research grant on the drug screening. And the Harry Orr lab has recently come on board and developed this SCA5 mouse model. And we've had support from NIH and also more recently, National Ataxia Foundation. And I'll stop there.
Thank you, Dr. Avery, for explaining SCA5 to us, as well as sharing some of your own research. We very much appreciate it. We have time for a few questions. If you wouldn't mind stopping your screen share, just so that people can see us a little bit easier. Fantastic. So we'll jump into questions. So the first is about MRI. Are people able to diagnose SCA5 just from the MRI? Are you able to differentiate between ataxia types?
That is not easy for me to answer. I can say that originally SCA5 was considered a pure cerebellar disease, so you would not expect to see defects in the brainstem in an SCA5 patient.
Mm-hmm.
But that's not really my, I think a clinician that looks at these MRIs for other different SCA5 patients would be able to better answer that. So I really am hesitant to try to say anything more than that.
That is a-okay. So we'll ask folks to rewatch Dr. Z's talk. She talks about this a little bit, if I remember correctly, about some of the conditions that SCA5 can be mistaken for just on the MRI, which is why you really need a genetic test in order to get that confirmed diagnosis. So our next question from an audience member, uh, what is the evolutionary advantage of SCA5, which causes the cerebellum to deteriorate? Shouldn't having an inherited condition like this have died out thousands of years ago? Is there anything we know about this?
So that's a good question. It's many SCA5 mutations are late onset and people are living longer. So it could be that SCA5 has persisted in the population because you don't have any symptoms at the time you're having kids. Or if you weren't living as long 100 years ago, you never developed symptoms. So that's, I think, for the late onset mutations. For the early onset mutations, I think it's a different case where if you have an early onset mutation, in that case, if you know you have this disorder early on, you may choose not to have kids later. So
that makes a whole lot of sense. Next, we have a question from an attendee whose child has a de novo mutation in SBTBN2. And they've been told that this mutation may cause SCA5, it may cause SCAR14. Why is it that sometimes geneticists are unable to tell the difference of if this is SCA5 or SCAR14?
okay, so SCAR14 in general, there's a couple of things here. SCAR14 is a recessive disorder. So if you have SCAR14, that means you have a mutation in each of the beta SBTBN2 genes. It doesn't have to be the same mutation, but you would have a mutation in both. And those SCAR14 mutations are typically loss of function mutations, and a common type of mutation that results in loss of function is a truncating mutation, where either a stop codon has been introduced, so that could be a single base change, but it leads to a premature truncation of the protein. Another possibility is that there's a deletion of bases that results in a frame shift. Frame shift mutations often lead to the introduction of a stop codon. And so often these truncated proteins are not stable, and that leads to a decrease in protein expression. So it's considered would be considered a loss of function mutation. So SCAR14 is associated really with the loss of function mutations, like truncated mutation. If you have a single mutation and it's not you just have a mutation in one of the SBTBN2 genes, then that mutation, if it's causing disease, is causing disease by a dominant effect, which is, so it's not a recessive mutation. And but it can still result in symptoms that are similar to SCAR14, because if that dominant mutation is causing early onset SCA5, the symptoms of early onset SCA5, at least as I understand them, I've read, are similar to SCAR14, which are more severe early onset symptoms. But again, I just want to say I'm not a neurologist, and I'm not seen doing clinical studies or tests on these patients in a clinic.
Well, thank you for clarifying that and explaining the difference between these kinds of mutations. We have time for two more questions. The first is, a couple of people have asked this. You talked about the different approaches that are being taken to develop therapeutics for SCA5. Is there, from your knowledge about what's being done, is there going to be differences in the medications that are developed for adults versus children? Or is it going to be the same kind of medication that will just be tested in two different populations?
That's a tricky question to answer because medication for kids is different than medication for adults.
This is true.
I think that is not something I've thought so much about. I have been interested. So for example, the SCA5 mutations in the ABD that I've characterized, about half of those are associated with early onset and about half are late onset. In my mind, if we can reduce binding of that mutant beta spectrin to actin, it's going to be beneficial regardless if it's a kid, child, or an adult. But I have not gotten to the point, at least in my thinking, this drug discovery really takes a team of people with different expertise. And so I have not thought about some, I don't know, challenges of using the same compound in a child as an adult.
Well, thanks for letting us know. And then our last question today is a clarification, as you shared some data at the end of compounds you're going to be testing in additional mouse models, looking at different side groups. We have someone asking here, why can't those compounds just be tested in humans right away? Why does this additional work need to be done?
Because they haven't been tested. All right, so one is a mycofungin. That's an antifungal. Usually antifungal, it's an FDA approved antifungal. You are wiping that as a cream maybe on your skin to treat an antifungal. SCA5 is not a skin disease. And so you would have to inject it basically, or get it somehow into the cerebellum. And to target beta-III-spectrin, but and maybe it would work, but you would probably have to take this drug for a long time, a continuous period of time, and while this compound has been injected for systemic infections, that's more an acute use of the compound. And so there's been no studies, for example, that have so we don't know what concentration of the compound that would be needed. It's possible that you need a very high concentration of the compound to have any beneficial effect. That would require a clinical study to see if it's safe to use that compound at a high concentration without having any serious side effects. The second thing is we're still characterizing mechanism of action for all of those compounds. We used an assay that had just two proteins in it, actin and beta-spectrin. The actin subunit is about the same, a single actin molecule is about the same mass as the beta-spectrin ABD. So in my mind, I think it's equally likely that mycofungin compound is causing reduced actin binding by targeting beta-spectrin or targeting actin filament. You do not want a compound that is targeting the actin filament because lots of different proteins bind actin, and you don't want to disrupt actin function in general. We want to target beta-III-spectrin specifically. So there's some more characterization of these compounds that need to be performed to better understand them, to optimize them so that we can use them at lower concentrations. Maybe to, in the case of mycofungin, it's a very large compound. It'd be better if we could reduce its size. So you could take it orally and maybe it could be across the blood-brain barrier. So it would be really dangerous to take any of those compounds right now as thinking that they're going to help you.
So what I'm hearing is, although these results are promising, they're very early days, and in order to have a better sense of safety, we need to have a better understanding of what the drug exactly is doing in the brain.
Now, that doesn't mean that we don't inject them in a mouse.
Exactly.
Knowing a little bit more, because we want to show efficacy. We want to show that this therapeutic approach to target reduced binding of mutant spectrin to actin actually can be useful as a therapeutic, but that's in mouse. All right. And
yeah, baby steps.
Baby steps. So I want the work to go as fast as possible, but it does need to be safe, and it does meet. It's hard to cut any corners.
Well, it makes a whole lot of sense. Thank you so much, Dr. Avery, for sharing your time and expertise with us. We truly appreciate it. Thank you so much for joining us today.
Thank you.
And thanks to everyone who attended. I know we went a little bit over time, but I hope you enjoyed this talk and you learned something new about SCA5. So wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, everyone.